introduce.
Okay, so the live streaming recording on.
Okay. So welcome everybody to our monthly, but actually this month there are going to be two colloquia in biophysics. We have the privilege of listening to Dr. Serah Kimani this afternoon. She completed her master's in structural biology and a PhD in molecular and cell biology at the University of Cape Town under my supervision. I must admit, it was an amazing experience teaching her and working with her. So she conducted research on the catalytic mechanism of the amidases, which was really significant. And after that, she undertook a postdoctoral research fellowship in the Department of Medical Cell Biology under the guidance of Professor Sharon Prince. And she focused on the discovery of anti-cancer compounds. She then moved to Toronto in Canada and spent the last 5 years at the Structural Genomics Consortium studying E3 ubiquitin ligases and their applications in targeted protein degradation. Now, this afternoon, she's going to talk to us about PROTACs, I guess, which are things that multi-proteolysis-targeting chimeras. And she's going to talk about this protein, DCAF1, that plays a critical role in protein homeostasis. So she's still in cancer, and she's been using artificial intelligence to figure out ways of preventing cancer. And I think that's all that I'm going to say now, and she can talk about the rest. So over to you, Sarah. You there?
Yes. Can you see it?
Okay. Yes, I can see.
Yes,
I can see.
but we can barely hear you.
But you've gone very squeaky, Sarah.
Squeaky? Okay,
Yeah, there you are. There you're fine. Okay, you should be good now.
Let me see if I can. Maybe the volume. I'm using a new laptop.
No, but it's perfect now.
Okay.
Yeah, as long as you speak like that, that's perfect.
Okay.
But you need to make full screen again. You were
Yes.
full screen and now you're not full screen.
Yes.
Yeah.
Now I'm full screen.
Now you are. Yes.
Okay, so hi everyone. Thank you, Trevor, for the introduction and for invitation to talk about let me hide this. Okay. Yeah, and for the invitation to talk about some of the work that I was involved in, the Structural Genomic Consortium, which has been part of the University of Toronto. So today I'm going to be telling you the story of how we found DCAF1 ligands and how we found DCAF1 ligands using AI-driven technologies, and how we used structure-based advancement of these ligands to make PROTACs and chemical probes. But before I actually get into the talk, I want to talk a little bit about Structural Genomic Consortium. So this is a charitable research organization that has 6 sites around the world. And I was based at the one at the University of Toronto. So Structural Genomic Consortium is dedicated to open science and has for many years collaborated with so many different organizations within the public and the private sector. So as maybe if some of you might know, that Structural Genomic Consortium was actually started as as a program for determining as many structures in the genome as possible. But as time went by, they've changed their focus, obviously, with especially recently with the invention of AlphaFold and all that. They've changed their efforts not just to look at the structures themselves, but to also develop bioactive compounds. And they actually have a very, a very yeah, they have a target. They have a very ambitious target to find ligands for every unliganded protein in the human genome by 2035, which is called the target 2035. So yeah, so they've really embraced this data-driven chemical biology. They've been mobilizing community. They have this competition that is called CAS Challenge, where people people all over the world actually can participate to use computational methods to find ligands that are then purchased. Whatever they find is then purchased, and it's tested at the LGC labs. And that's one of the ways that they're doing, but they're also using these targeted high throughput screening technologies and machine learning to try and do this. So at the SGC, when I joined SGC in 2019 when I left South Africa, and I've been involved with quite a number of different types of proteins, but mostly my work has involved E3 ligases, trying to look at how we can find the ligands for chemical probe development and also for targeted protein degradation. So I've also worked with a few other proteins, but my main focus of my work was on E3 ligases. Some of them are shown here, and also components of them like DCAF1 and DCAF11. And I've had the privilege of working with so many collaborators, both in the industry and also in academia. So yeah. So now I'll get into the topic of today, talk about targeting the DCAF1 WDR domain. This is work that we started during COVID, and I'm privileged to have been there from the beginning of this project, and I was tasked with the responsibility of putting together the protein infrastructure that was used both for screening and for structural work, and also eventually I ended up leading the protein biochemistry and biophysics and also structural studies of this program. So as you will see, it was a very highly collaborative project, as you will see as I go on. But it was mainly led by Structural Genomics Consortium, and later the Ontario Institute of Cancer Research joined. And it's been amazing to see the progress that a huge collaboration like this can make in a short period of time. So yeah, so DCAF1 is a protein that is involved in ubiquitination pathway. So I'm going to first set up the stage so that we are all on the same page and talk about a little bit about ubiquitination. So this is a post-translational modification that has diverse roles in the cell, and it involves covalent attachment of ubiquitin, which is a 76 amino acid protein on a target protein. And so the ubiquitination is actually a cascade. It involves activation of ubiquitin by E1 enzyme, and then conjugation to E2 enzymes. And then what happens is that then we have these E3 ligase proteins. And in this case, I'm showing a complex. So when we have this ring-type E3 ligase complex, the E2 ubiquitin complex actually binds to the E3 ligase. The protein that is going to be ubiquitinated, which is here called the substrate, also binds to the E3 ligase complex on the other side, and this allows ubiquitination of the target protein. And like I said, the ubiquitination has so many different roles in the cell, but in this case, but one of the main studied and well-known consequences of ubiquitination is proteasomal degradation, where based on where the ubiquitin is attached and how it's elongated, the protein is actually degraded by the proteasome, and this actually results in the recovery of ubiquitin, recycling of ubiquitin. So the protein degradation system has been harnessed in kind of a growing therapeutic modality that is called targeted protein degradation. So this is where E3 ligases in the cell hijacked, or you make some compounds that hijack them to degrade pathogenic proteins that you don't want. It's a very powerful method because it can be used for proteins that cannot, that have previously not been able to be targeted by inhibitors. So one of the ways that this targeted protein degradation is achieved is through PROTAC molecules, which are proteolysis-targeted chimeras. So these molecules will have a ligand for the target protein. So you'll have a ligand for the target protein, and you will have a ligand for the E3 ligases, and then you will have a linker in between. And so what happens is that in the cell, I'm trying to remove something. Okay. Yeah. So what happens that in the cell that you can get this PROTAC now to bind to your E3 ligase of choice, and then it will bind to the target protein that you want to degrade. And so this leads to polyubiquitination of the target protein and eventual degradation. So I think what is really interesting with which is really amazing is that with DCAF1 being in this pathway is that as a protein, as you will see, it can be targeted itself for E3 ligase complex. So yeah, and this is just a note that there are over 600 human E3 ligases, and only a very small percentage of them have actually been recruited or have actually been used in targeted protein degradation. And a lot of times people have seen that resistance is normally observed with commonly exploited E3 ligases like VHL and CERBN. And so more E3 ligase handles are needed. So just to talk to you a little bit about the human DCAF1 protein, this protein is one of the many DDB1 CAL4 associated factors that serve as substrate recruitment modules for E3 ligases in the cell. And specifically for DCAF1, it serves as a substrate recruitment module for two E3 ubiquitin ligase complexes that are shown here, the RING-type CRL4 and the HECT-type EDVP. So basically, DCAF1 will be found attached to DDB1, and then it will recruit a substrate. And then as I explained earlier, when the E2 ubiquitin complex binds to this E3 ligase complex, the substrate is ubiquitinated and polyubiquitinated, and based on the function, if one of the roles of ubiquitination is proteolysis, then the substrate is degraded. So out of the over 1,500 amino acids of DCAF1 protein, the C-terminus encodes a protein-protein interaction module called the WDR5. So this module folds into a typical 7-bladed beta-propeller WD40 repeat domain with an overall donut shape. And as you can see here in the structure, so looking from the top, you can see the donut shape with a central tunnel in the middle. And from the side, this is how it looks like. And one of the blades, so the reason why these proteins are called WD40 is that one of the blades is formed by around 40 amino acids, a small structural motif of around 40 amino acids that terminate with W and D, which is why they're called WD40. So when these donuts binds to DDB1 in the complexes, the peptides of interacting keep removing them. The peptides of interacting substrates can bind either in the central pole, which for all the proteins in this family is very, very diverse. So they can bind either in a donut hole, they can bind at the top here, they can bind on the side of the donut. So DCAF1 was actually discovered as a cellular target of HIV accessory protein R. And so it's also called the VPR binding protein. So what happens is that through the WDR domain, the VPR proteins bind to the DCAF1 WDR domain. And so in this way, they hijack the whole E3 ligase complex, both the CRL4 and the EDVP. They hijack them to promote proteolysis of cellular protective factors, like the one that I'm showing here, SAMHD1. So this protein is very important because it blocks replication of HIV in immune cells. And so what happens is that, as shown in this crystal structure, we have the WDR domain, the VPR protein binds at the top of the donut, and then it recruits, so this is just a peptide, but the SAMHD1 is a big protein. It recruits SAMHD1 and other proteins, which now get ubiquitinated and they get degraded. So this way HIV can actually continue replicating in the cell. So for this reason, DCAF1 can be seen as a very interesting target for protein-protein interaction inhibitor design. Physiologically, the DCAF1 is associated with so many different processes, but one of the most interesting one is its role in cell proliferation, because it's been shown to lead to tumorigenesis. And even though, as you can see, this is a review paper, as you can see, there's so many questions, there's so many things that are still not understood, like how it actually does what it does. So the molecular function is still under investigation. There's some studies that have proposed that DCAF1 actually promotes degradation of the tumor suppressor proteins. And that is how it, in some cases where it's overexpressed, it leads to tumorigenesis. So based on the little background that I've given you, we see that DCAF1 is an interesting chemical biology target. So it can be used for PPI inhibitors, protein-protein interaction inhibitors. So in the case of the story of DCAF1 WDR being hijacked by lentiviral VPR, so it can be used for inhibitors that actually disrupt its interaction with the lentiviral proteins. And in the case where DCAF1 is used to possibly degrade tumor suppressor proteins, we can find inhibitors that can disrupt its interaction with these proteins. Most importantly, DCAF1 can also be used in targeted protein degradation. And so it can be used as a handle where we make PROTACs or maybe molecular glues or anything else that degrades proteins that we don't want in the cell, pathogenic proteins, but in itself, it's been shown to be overexpressed in some cancers. So in that case, we could actually make PROTACs or other, targeted protein degradation modalities where we now have other E3 ligases actually degrading DCAF1 on its own. And so, as I mentioned previously, the molecular function of this protein is still under investigation. And so, apart from all this, we also would need a good chemical probe. So this is a cell-active protein that can be used to study the protein. And also, that can be used to study the function of the protein. But this protein has previously been classified among the big percentage of proteins that are undruggable. And so in this study, we used two AI mediated technologies to try and find ligands for DCAF1. It's not a protein. It doesn't have any nice pockets that can be targeted. So these methods were actually really, powerful in finding binders for this protein. So one of the methods that we used is this drug target interaction prediction method. So we collaborated with a company called Cyclica that has long been acquired by another company called Recursion. So they use proteome-wide proteome-wide. They have made this matchmaker software that is a deep learning engine for proteome-wide prediction of small molecule target interactions. So basically, what they've done is that they have generated this huge pocketome of a whole lot of proteins that they could find in the PDB, and I'm sure they've now have a huge pocketome of a lot of proteins that have actually been predicted by AlphaFold probably. And so they will try and find, so basically without going into details, is that they will try and find a compound that is binding to a protein of interest and that is not binding to all the other proteins or the other pocketomes that they have in their database. So after their experiment, and they also used molecular docking and property prediction and molecular profiling. That helped them narrow down the amount of compounds that they found. So what they did is that they selected 100 compounds that they sent to our lab for SPR evaluation. So SPR is an optical method that actually determines compound binding in real time. So the protein is attached to a chip, and then the compounds are flowed on the chip. And what happens is that there's resonance signal that is generated when the compound is binding. So this method is very sensitive. It's amenable to high throughput, doesn't need labeling of the protein or anything like that. So it's quite a good protein. It's quite a good method for what we're trying to do. So basically what happened is that out of the 100 selected compounds that we received from Cyclica, we found one compound that bound to DCAF1 with very low affinity of 70 micromolar, but it was a binder. And we tested it against another WDR protein called WDR5, and you can see good selectivity of this compound. So we went ahead and we determined the structure of the DCAF1 WDR domain with this compound. And this is where, having a very good protein system actually really helped for us to see what was happening in this compound. So, and you can see we got 1.5 angstrom structure, and you can see the density of the compound. The compound binds at the top of the central tunnel of the DCAF1 donut. And looking from the side, you can see where it's binding. And when we actually aligned our structure with the structures of DCAF1 that have VPR aligned, you can see the compound actually is in the pocket where actually VPR binds, and this is a close-up picture of that. So this was very encouraging. And although we did not pursue this compound to try and make it better, other people, our collaborators from Merck, have actually this compound and have improved on this compound, and they've then made PROTAC molecules that are enabling degradation of various proteins that they are characterizing at the moment. So for us, we just went ahead and published this as a methods paper. And then, so this was the paper. So then I'm going to show you, and the reason why we did not actually proceed with this is because we got better compounds using another method. So in the other method, we used an affinity selection method called DEL. So it's DNA encoded library screening. So there are these companies that make these huge libraries of compounds, billions and billions of compound libraries. And each compound is actually tagged to a unique nucleotide. And so what happens is that you immobilize your protein. So when you make your protein, it needs to have a tag for whatever surface you want to immobilize it. And then you incubate the protein with this huge whatever library of compounds. And then you wash the non-binders. And then what you do is that to check whatever bound, you do DNA amplification, and then you sequence the DNA, and that way you'll be able to figure out what actually bound to your protein. So obviously, with this method, you will then go ahead and do experimental evaluation and binding visualization. So with this method, we collaborated with a company called XChem that has huge libraries of these DEL compounds. And so what happened is that after they ran DEL, whatever output they got was used by another company that we collaborated with called Zebi AI. So this is it was an AI company actually that was bought, that was after this project, or after the success of our project, it was bought by another company called Relay Therapeutics. So what Zebi AI did is that they used the output from DEL, they trained AI models, and then using the trained AI models, they were able to score enamine compound libraries, like libraries that the commercial libraries like enamine. So from there, they were able to identify compounds from the library that we could directly purchase, that we could directly buy and test in the lab without having to try and synthesize whatever was found from DEL. So that was very powerful in the sense that we could now test a whole lot of compounds to try and find, leads. And like I said, SPI is a very good method for high throughput screening. So so initially we got 33 ml predictions from Zebi AI, and then we tested them with SPR. Whatever data we got, we sent back to Zebi AI, both positive and negative. They used that to train the models and to enhance the model so that it can know the false negatives and what were the false negatives and what was the positives. And so what happens is that they sent us through rounds of optimization, they sent us around 600 compounds. And so from there, we were able to identify one nice compound that had SPR KD of 11.5 micromolar. So we went ahead and we determined the structure of this compound. And so we found that the compound binds. It also binds in the central tunnel. But then what was interesting, looking from the side, is that you see actually the compounds bind deep into the central tunnel. And so we started thinking about how we can improve this compound and how we can, extend it to the surface so that it can be useful for maybe PROTAC development or whatever. So what we did is that our computational guys, collaborators from OICR, actually looked at the structure. So anyway, before I go there, I'll say that the ML model predicted an R enantiomer of the compound, and this is what we actually purchased from enamine for testing. But then when the computational guys looked at the crystal structure, they found that the R enantiomer had a suboptimal geometry around the central amide, which is here. And when they did molecular modeling, they found that actually the S enantiomer fits much better within the crystal structure. So, but when we looked at the density, because for some reason this structure was only 2.3 angstrom resolution, we really could not tell, whether it was R, whether it was S enantiomer. So we went ahead and synthesized, pure enantiomers, both R and S. And what was interesting is that we found actually the S was actually a more potent binder of DCAF1 WDR protein with a SPR KD of 0.5 micromolar. So we solved the structure, and you can see, very good density, and we got decent resolution, and you can see the compound in the pocket. So what we did is that we went back and we did chemical analysis of what we had actually purchased from Enamine. It was supposed to be an R enantiomer. It turns out that it had a 4% S enantiomer contamination, and this is probably what bound to the crystal structure. But because we tried to force an R enantiomer, and that is why the computational people actually found that the S enantiomer was actually a better fit. Anyway, so we went ahead and did limited SAR of this compound, basically substituting these two rings. And what we found is that we improved the binding affinity to 38 nanomolar, and we went ahead and solved the structure, and we could see how the extra groups were actually interacting with the protein. So then we went ahead and looked at the cellular engagement of this compound with DCAF1 WDR protein. So we used thermoshift assay called CETSA, and we could see very potent engagement of the compound with the DCAF1 protein. So at this stage, we published this our lead compound, and so this has actually been quite an amazing thing to watch, because a lot of people have now used these compounds to design whatever thing they want, and it's yeah. Anyway, so from there we wanted to see how we can optimize this compound. As I showed you, it binds deep in the pocket, so we wanted to see how we can extend it to the surface. So first we looked at substituting this ring, and so we tried to make an N-methyl. We just tried to substitute with a methyl group, so we made an N-methyl pyrazole, but this actually killed the potency of the compound. So what we did then, we replaced the pyrazole with a pyro group, and this allowed us to actually add substitutions to this group. So when we changed the pyrazole to pyro, we actually lost affinity. So we came from like maybe less than 100 nanomolar to like 700, but then substitution on the pyro actually seemed to restore the affinity. So in this case, we added an alkene group, and we did this because we hope that the alkene group will be positioned like straight out of the pocket. And so we solved this crystal structure, and for sure the crystal structure showed us that the alkene groups point straight out of the pocket. And it also showed us that the change of pyrazole to pyro actually retains these water-mediated hydrogen bond. So from there, we wanted to do more optimization. So more optimization of this compound actually led us to two compounds. So one of the compound had these alpha beta unsaturated amide, and the other one had these pyrazole group. And we solved the structure, and we saw that both groups were actually very well tolerated in the structure. So from here, we decided we wanted to go two ways. We wanted to see, can we make a chemical probe, like a very potent compound that is cell accessible, that can be used to probe the function of DCAF1, or can we make PROTAC molecules that can actually recruit DCAF1 for degradation of other proteins? So I'll first show you what we did with the chemical probe part of that. So from our two compounds, the one that I said, one has the alpha beta unsaturated amide, and the other one with the pyrazole. So, and these are the structures. Each one of them was optimized, and we came up with two compounds that were very potent binders. You can see one of them had a an SPR KD of around 2 nanomolar, and the other one had an SPR KD of around 8 nanomolar. So this one has a piperidine group, and this one has a morpholin group. So we went ahead and looked at the structures of our two best compounds, and you can see they're very nice density, very good structures with decent resolution. And here are the surface representation showing you the compounds aligned with our parent hit. And as you can see how the two compounds actually, were extended to the surface. And so when we align the two structures with the two compounds, you can actually see how the exit vector is different for the two of them, despite them starting from the same point. And I mean, the pocket kind of gets wide at the top, and that is why it was able to accommodate these two diverse conformations. So in terms of the chemical probe, we now needed to figure out whether one of the compounds that we've made actually meets the criteria of the chemical probe, and these are the criteria for that. So the compound needs to have biochemical potency of less than 100 nanomolar, cellular potency of less than 1 micromolar. It needs to be profiled as much as possible, needs to have good selectivity, and it needs to show a proof of target engagement. And these are desirable, like it shouldn't have a pains element that can interfere with assays. And then we need to have availability of an inactive control. That is very important. And then obviously the data and reagents being available for other researchers who want to use, and then being soluble and being stable. So yeah, so just for a more detailed, deeper definition, a chemical probe is a small molecule designed to selectively bind to a specific protein target and modulate its activity. And this allows study and understanding of the protein function in the biological system. So looking at our two best compounds that I showed you earlier, I won't go into details, but I will show you that we finally selected, trying to remove this. Yeah, we finally selected OICR 41103 as the best compound that met the chemical probe criteria. You can see in terms of in vitro potency, it has a KD of 2 nanomolar. And then it was shown using two different methods in the cell that it actually engages with a WDR domain of DCAF1. It has no cytotoxicity in these cells up to 10 micromolar, which is important because when researchers are trying to use it, you don't want the compound to be killing the cells when you're trying to use it. You want to have room for you to be able to use compound concentrations, so you're able to look at what you're looking for in the cells without the cells dying. And then in vitro, we did selectivity as we used SPR, and we looked at the compound binding to these 6 other WDR proteins, and we found no binding to these WDR proteins. And then we developed a control, which was an enantiomer that had different binding capabilities to the actual compound. So this chemical probe is now available in the chemical probe library, and we recommended a dose, especially in the cell, of 1 micromolar. So anybody who would want to study DCAF1, see what it's in the cell, they can order this compound, and it will be available together with all the reagents that are needed. So anyway, so I went ahead and looked at the structure more closely, basically just aligning the structure with the DCAF1 structure that has VPR bound. And as you can see, the protein actually binds the compound, the end of the compound actually binds to where the VPR protein, which is here in green, is binding. And so we went ahead and did both in vitro VPR displacement assay and in vivo. And as you can see, the compound is potently able to displace the VPR from the protein, both in vitro and in vivo. And so this was really promising. This was really nice because then we have now a compound that is able to perturb, the activity of DCAF1, at least with regard to its binding to a VPR. And hopefully this compound could actually be developed into a VPR DCAF1 PPI. So that's the story with the chemical probe. Now I'll tell you a little bit about what we did to get to in regard to targeted protein degradation. So we wanted to see if we can make PROTACs that can enable degradation of human WDR protein. We chose this protein because it's a protein we had worked with a lot in the lab, and it's a protein we know very well. And so this protein is also a WDR protein, and it functions as a scaffolding protein in gene transcription. It interacts with oncogenic transcription factor MYC and MML, methyltransferase, and its overexpression is implicated in various cancers, including bladder, breast, and leukemias. So it was a good candidate for us to see if we can be able to use DCAF1 whatever PROTACs, DCAF1-based PROTACs to degrade. So, and this is just to show you the schematic. So we used some of our best compounds at DCAF1 handle, and then we selected a published WDR5 ligand, and then obviously we needed to find a linker to link the two, and to try and see if we are able to actually degrade to enable degradation of WDR5. So, and here is so we selected this ligand that was actually developed by OICR and has been used in the VHL-based degraders in this publication. And then we used two of our best, like I said, with different substituents here. So we used those two to make ligands. And so the main thing was to find linkers. So we started with simple alkyl and PEG-based linkers. And with basically from having between 4 to like 16 or 20 atoms. And very quickly we could see that the shorter linkers were not able to enable the formation of a ternary complex. So we were just using like SPR to try and see, are we forming a ternary complex between DCAF1, the PROTAC, and WDR5? So what we did, so that actually informed the, the design of, second generation of PROTACs. And so eventually, we had these 4 PROTACs. You can see they are all using the PEG linkers, but different lengths. Some of them, one of them, I think, is using this compound, and the others are using this. But yeah, but what is important is looking at the cellular degradation of WDR5. And so, I'm just trying to find. So in this case, this is a cellular assay that was done by Mohammed from OICR. And so he managed to determine the Dmax, which is the percentage of protein clearance observed in the cell, and the DC50 is like IC50, is the concentration required to clear half of the Dmax. And as you can see, based on the length of the linker, you can see as the linker gets bigger, so this one has 9 atoms here. So you can see as the linker gets bigger, you get more potent degradation of WDR5 in the cell. So that was really, really encouraging. And so we went ahead and saw some of the crystal structures of the WDR, WDR5 DCAF1 with the PROTAC, and this is with PROTAC4, which was, as I showed in the previous page, was the best. And so, yeah, and so here I've aligned a structure of CRL4 DCAF1 E3 ligase. And so this is our structure here. So aligned with our structure here. And you can see how nicely, if we had E2 protein here, you can see how nicely WDR5 is actually poised for polyubiquitination. And so this explains why we were able to see that that potent polyubiquitination with this polyubiquitination and degradation with this PROTAC. So I believe OICR and SGC have gone ahead and developed, second generation WDR degraded PROTACs, obviously trying to improve the PROTAC to make it like a drug-like. So like looking at the molecular weight, the surface area, the hydrogen bond donors, reducing the amount of hydrogen bonders and reducing maybe the amount of rotatable bonds. So, and I think this is work that is still ongoing. So basically for this, we just went ahead and published a quick crystal structure just to show the ternary complex and to actually try and predict like how the DCAF1 actually recognizes its substrates, because there's no information out there. So basically, in summary, I have shown you how we have used 2 AI-driven technologies to find DCAF1 binders. One of them is the drug target interaction ML model, in which case we found an SPR a compound that has SPR KD of 17 micromolar. We solved the structure and we found this compound actually binds where VPR lentiviral VPR proteins bind. And so, and Merck, our collaborators in Merck have taken this compound and made some PROTACs that they actually now characterizing that are able to degrade various proteins. And then we used the DEL screening technology combined with machine learning. And these we were able to find a compound that was optimized to, that was structurally optimized to a KD of 38 nanomolar. And then following further structure-guided development, we were able to develop DCAF1 chemical probe that actually is able to displace VPR from DCAF1 and also to develop PROTAC molecules, DCAF1-based PROTAC molecules that are able to degrade, to effect degradation of WDR5. So, and based on the success of DCAF1, the initial success, we decided to now look at the WDR proteins in the whole genome. So the human genome encodes around 350 WDR proteins. So we selected 249 based on various criteria. And from those, we were able to purify 95. And so we did DEL screening of 16 of them, including DCAF1. And we found, we were able to find ligands for 7 of these proteins. And you can see with KDs from below 10 micromolar and above 20 micromolar. And we were able to solve, some of the structures of these compounds together with the respective protein. So yeah, so that was very encouraging. So we published a paper which was called Target Class Ligandability Evaluation of the WD40 repeating containing proteins. As you can see, there's so many people involved because it was such a huge project working with all these proteins. We started with 249, like I said, ended up with 95, and then we selected 16. And then Yeah. So just to wrap up, I wanted to show you these other affinity selection method that SGC has adopted and they have developed in-house. So this is called affinity selection mass spectrometry, and they have purchased these high resolution mass spectrometry machine, and they have also instituted like ML training. So in order to be able to look at the compounds that they get from ASMS and then try and train models that can go into the commercial databases, find compounds, similar compounds, and get those compounds. And so they're able to buy those compounds and get them tested in the cell. So in this method, you just have your protein, you incubate it with compounds, you wash off unbound compounds, and then you denature the protein, and then you determine whatever bound using high resolution mass spectrometry. So this protein obviously requires compound availability of compound libraries, and it also requires the protein to be highly pure, because you might get compounds that are binding to your contaminants. So this is one of the criteria, that your protein needs to be highly pure. So SGC has assembled a huge compound library already from industry and academic partners, and then there's an acceleration consortium that is using self-driving labs to synthesize a whole lot of compounds. So that's like really ongoing, and this technology has actually been applied to some of my targets, which were like these E3 ligases. They are all E3 ligases. So that's very interesting. And just to summarize and to just have final comments about, how we see, AI, contributing to accelerated hit discovery for traditionally undruggable proteins. Technically, the DCAF1 and all the WDR proteins that we selected were classified as undruggable because they're not enzymes, they don't have nice pockets that can be targeted. And so these methods have actually allowed us to find compounds for proteins that otherwise did not have any binders. And obviously the use of these affinity selection methods to the DEL. Obviously, DEL is an expensive method because the companies that make them sell those libraries, but these affinity selection mass spectrometry is a method that can be implemented easily in the lab. But the most important thing is the use of machine learning on the compounds that are found here to find compounds in the commercial libraries that you can actually just purchase and select and test in the lab without having to go through the synthesis process. And obviously in these projects that I presented, the structure-guided heat advancement, having those structures and seeing how the proteins how the compounds are actually binding to the protein was very useful for us to be able to advance and make chemical probes and PROTACs that I showed you. And so here are a whole lot of people that are involved in the work that I showed, different companies. So obviously there's SGC, there's OICR that I see were leading the project. There's ZBAI and who did the ML training of the DEL. There's XChem who did the DEL screening. There's Cyclica who did the DTR method. There's Mark who advanced one of our low affinity compounds, and these Pyramol who actually synthesize compounds. Yeah, so whatever we cannot synthesize in-house, we would contract them to synthesize. So yeah, and then I just thought I would put here these here for people who are not on the slide. And so I'll leave you with those pictures that I took recently on my adventures in our ongoing fall, autumn season, which for me, I find, I mean, I used to really like flowers and all that, and I still do, but I find the autumn season being so amazing that trees can actually burst out in these amazing colors, just before they go to sleep in winter. So it's really amazing. I hope this still works. If you want to connect, we can connect on LinkedIn, and these are my email addresses, and I will leave this. Sorry, my phone is ringing. Yeah, so I will leave that. I will leave it at that, and I'm happy to answer any questions.
Thank you very much, Sarah. That was a beautiful talk. I do have a whole, well, I have a question that I'm going to take my chairman's privilege and ask first. When
Yes.
you do this machine learning, what are the inputs and what are the outputs of the machine learning? What do you put into the program and what do you get out?
So what you put in, so the machine learning, what you put in is the compounds, the compounds that were identified from Dell or from ASMS. So you put those in, and then what you do is that the ML model, you use now the ML model. So you use them to train an ML model. So you use those to train, and then what you do is that you take that ML model and you query public commercial databases. So the good thing with that is that, like, if you had, like, let's say 100 molecules or 600 molecules identified from Dell, it would be so hard for you to actually synthesize all those and figure, because Dell will just tell you these are the compounds. And it will be so hard for you to be able to synthesize all those. So what the ML is doing is actually, it's like an intermediate process. So it goes to the commercially available databases, and it looks at the compounds that are very similar or the same to the ones that you found using Dell. And so from that perspective, you can now order those compounds and test them in the lab. And the beauty of it is that once you test those in the lab, whatever results you get, you can input them back to the model. So now you know which one works, which are positive, which are negative. And so you optimize the model so that when it goes now to the commercial databases, it will actually be able to discriminate the good and the bad. So the good, like the binders and the non-binders. So that's how, that step actually accelerates the whole process.
Does the user, like your people, do
Yeah.
they have to actually train the model to find the compounds for your specific protein? So you train it to recognize binders for a specific protein, do you?
So you train it for that specific protein. So what happens is that after you train, the model doesn't go back to the protein. The model actually just goes to the chemical libraries, looks there, sees what are the compounds that actually look like this or have features that are in these compounds. So it will just give you more compounds. And then what you get is like a list of compounds that you can order from Enamine or other libraries. And then you go back to the lab and test them using SPR. So the model, the model where we are training using Dell data does not go back to the protein.
Okay.
Yeah.
Okay. So that's enough for me then. Thank you, Sarah. Does anybody else want to ask a question? Are there any hands up? Okay, Lawrence, go ahead.
I was wondering if there are any dynamic events while the drug might be making its way from the pocket that you found to the surface of the protein. That is, are there any major conformational changes? And then going back to a previous talk that we heard, like I think last month before, could there be a ligand that sort of prevents that conformational shift and basically structurally locks the protein into place so that the substrate can't get in or out?
Or you mean, like, are there compounds that are blocking?
Yes, that block. Well, maybe not necessarily block the I guess what's the word? They don't block sterically,
Yeah.
but block conformationally, if that is a difference. That might be a distinction without a difference, really, but
Yeah, it's probably possible, but what is important is that this assess is done so many multiple times, and also what they do is that when they are doing screening, they actually partition, like they don't just do, or they don't just throw like a whole lot of them. It's like they partition their compound libraries so that you have small sub libraries that you're actually testing. And so, yeah, so that's what they do. And so I think with that way, you actually eliminate a big chance of having something that is blocking. But mind you that these compounds are not just binding in the pocket, in the central tunnel. That's just a tunnel, it can bind it. We're looking for binders that can bind anywhere, can bind on the surface, on the sides, because in this case, we're just looking for ligands that we can actually use to get E3 ligases and then combine them with the ligand for unwanted protein so that we can use them for targeted protein degradation. I don't know whether I answered your question.
Yeah, I think so. But let me ask a different question if I can, Trevor.
Yes.
So presumably the protein can be druggable or tagged for elimination if you raise an antibody against it.
Yeah.
Have you tried that one?
Yeah, so we're mostly focusing on small molecules, but obviously there are other groups like here in Canada, the companies that are here actually work on antibodies. So what they do is that if you can raise an antibody for a protein, then you can probably make antibody what's the word? You can do antibody drug conjugates, or you can actually try to harness antibodies for degradation. But we're basically, so that's possible, but we just focus on small molecules.
Understood. Thank you.
Yeah.
Jerry Joe, you had your hand up.
Yes, thank you, Sarah. Just a quick question. Is there any way you can infer binding site information from this experiment?
So from this, once you know the binder, then once you know the binder, the structures for us really help to knowing where the compound is binding and how it's binding. So, but if you don't have structures, I think it's kind of you're in a blind space. You're working in a blind space. You probably try and use maybe deletion methods and stuff like that to try and figure out. But for us, we had the advantage, and which is why I emphasize the importance of the structure-based advancement that we did, is that the structures actually managed to show us where the compound is binding. And that is why these methods that we're using, even affinity selection, I think eventually you need to go to the structure for you to be able to know where the compound is binding and for you to be able to advance. But without the structures, I think it's you probably can try and use maybe some kind of docking. But like I said, because they don't have pocket, some of these proteins don't have like a nice pocket. So it's probably very hard to figure out where the compound is binding. So for us, we had the advantage of structures. And that's how we were able to see where the compounds were binding.
Thank you.
Lawrence, you want another question?
I have another question about finding where the binding is on the surface. Have you done the calculation to map the electromagnetic field around the electric field, I should say, around the protein to see what kind of orbital steering and what electrostatic steering is possible around the protein?
Yeah, well, so we've done, we like in the beginning, we would do all that. So it's just data that I did not show. So in the beginning, when we don't, we will do all that because by the end of the day, you want to try and figure out where and all that. But like in our case, we didn't really focus on that so much because we had the structures and the structures were able to show us where the compound was binding in the end. So this is a case where like structure-based discovery is very central to the methods that we used in order to figure out where the compounds are binding and to advance these compounds. And even with the PROTACs, like I showed Even with the PROTACs, we didn't manage to solve structures of WDR DCAF1 with the PROTAC, because that then will show us how the orientation of the substrate actually affects ubiquitination and degradation.
When I was looking into this AI business for predicting binders, I came across a program called Boltz-2. Have you seen that one, Tara?
No, what did they show?
Well, they claim to be a competitor for AlphaFold, and has anybody in the audience heard of this Boltz-2? Sherry Joe?
No, I haven't.
Yeah.
Apparently, it's an open source version of AlphaFold, and it's been extended to look for binders. So
Yeah.
you can target any protein, and it'll tell you, it'll predict the structure, and it'll tell you where the binders are. Isn't
Wow.
that sound wonderful?
Sounds wonderful. Until you go and confirm. So what happened, I showed two methods, right? I showed the first one where they just used ML, where I talked about the cyclic collaboration, where they use the drug target interaction model. So what happened with them is that they tried to predict, because they kind of they saw where the compound, because they're kind of docking things, they can see where the compounds are binding from their ML model. But then what happened is that they predicted binding of that compound because it's binding to the surface, and the surface is so big. Where they predicted binding and how they predicted binding is not the way the crystal structure came out. So in the end, I think you still need to go to the experiment for you to actually get the correct picture.
That was a good thing to say. I'm glad you said it.
So that was so like for us, because we were like, can we see how the compound actually bound in their model before we actually did whatever? And they just say no, the model was not able to predict the collect orientation and even the actual place in that, because the pocket that the protein doesn't have a pocket, it's just like a very shallow surface. So it wasn't very good in doing that. And yeah. So I think with these promising models, I think you still have to go experimental and try and see exactly what is going on. And I mean, right now there are so many like AI things. So I attended another talk recently where people are actually doing phenotypic screening. So what you do is that you just treat your cells. If it's cancer cells, you treat them like I used to do during my postdocs. You find compounds, and then you train an ML model. It will go to whatever databases and find other compounds that are similar, and then you go back and try and test those and see if they're better or worse. But in the end, you still need to figure out what the target is, because you also want to see, is your compound selective, or is it killing so many different things? So yeah. So in the end, you still want to see the target, and I think you would still want to see where the compound is binding and how it's binding.
Okay. Does anybody else want to say anything? Well, with that, thanks again, Sarah. Thank you very much for a really stimulating and interesting talk.
Thank you very much. Thank you everyone for joining. Yeah, thank you.
Thank you. Thanks again. And thanks for joining, everybody. Bye-bye. For those that are left behind, I should actually say that we're having another talk next week. So don't be surprised if you get another advertisement.
Serah, congratulations.
Thank you.
We'll send an email. I'm curious to understand one thing.
Okay, yeah, send me an email. Keep me up.
Thank you so much.
Ooh,
Okay.
bye-bye.
Bye, Sarah. Thank you. Thank you very much. I'm going to end the meeting.